And so the question is, how do we decide these things in science? We do an experiment. And I think this is an experiment, if you had a school greenhouse, that you could do the experiment in with plants like brassica. I saw someone had brassica on their slide the other day, which is just cabbage, or with tragopogon, which is the example that I have. We would do an experiment and we would try to decide, is this new species really a new species? That's the first question we have to solve. And then there are other questions that turn up later. Now, what is that experiment going to look like? Well, we take two species of tragopogon. We'll take porifolius and dubius, interbreed them, and we get what I am calling a new species, Myrrhis. Now, if this is a new species, and if these are flowering plants, so there's no problem in even getting one member of the species to breed with itself, to propagate a new generation, taking that Myrrhis and now back-crossing it to the parental species, what happens then? Because if those back-crosses are also fertile, we would probably apply Occam's razor and say, well, if they're all interfertile, then they're really just one species. It would be like taking different breeds of cats, which are all interfertile, and we would only call that one species. The species idea has a certain privilege in biology. Now, if on the other hand those back-crosses are sterile, then indeed we do have a new species. So we can place our bets, do the experiment, look at the results, and we find that the back-crosses between the new species and the parent species are in fact sterile. Oh, this raises all sorts of questions, like, why don't we see this happening all the time? Yes, we haven't ever seen that before. Why doesn't it take over the whole range of the parents? What's the mechanism involved? How do we get a new gene pool into there? Isn't that what you're saying, if species have gene pools? And there are all sorts of questions like this that can be investigated in a scientific manner. This is not only about teaching how species originate, but teaching about how science operates. So let's take some of those questions. Why don't we see it happening all the time? Well, we do. That creationist canard that nobody ever saw a new species evolve is total rubbish. We do see them evolve all the time. It's just that we don't observe that speciation carefully enough to know about it in a lot of cases. Why don't they spread out and take over their parents' ranges? Well, they are in a hybrid zone. Both parent species are there, and they cannot out-compete the parents. That's the Darwinian idea that we have to bring in here. If there is a change in the environment, or if humans intervene to select out the new hybrid species, then there is a chance that these hybrid species will take over. And that is one of the patterns that we see in allopatric speciation, is that from a zone, peripheral zone, where there is some hybridization, that a change in the environment causes that hybrid to spread out over the whole zone. What's the mechanism? Where's the new gene pool coming from? Well, the mechanism is polyploidy. That is that the new hybrid species has twice the number of chromosomes than the original parent species. It combines the gene pools of both parent species. Now, if you look into how this works, it works in the first meiosis that doesn't complete with a cell division. And it could be said that none of biology makes sense except in the light of cell theory, and this is where cell theory can connect up very well with speciation and the teaching of evolution. If we understand meiosis correctly, then we can understand how this hybrid comes to a double set of chromosomes. First meiosis doesn't complete, the second one does, and those gametes are then diploid, they produce a tetraploid plant. We can then look at why the back crosses are sterile, and I was considering going around yesterday and asking people why bananas don't have seeds, because bananas are those type of plants, the seedless plants, which are the sterile back crosses of a new species to the parent species. They cannot complete meiosis, and the gametes die, and that's why they're seedless. What we are starting to see here is isolating mechanisms, and that's the idea from the modern synthesis. We could go back one step. we could go back to the animal hybrids and say, well, why are the animal hybrids sterile? And again, it's meiosis that's playing a role there. Now, some of these isolating mechanisms are going to be very strong, like the isolating mechanism in meiosis in the plant species. Others are going to be weaker. They're not going to result in full speciation the first time round. And eventually we can go along this path further and further, accumulating isolating mechanisms. And that we can start redefining a species as having accumulated enough isolating mechanisms over the course of time. Now, notice that along this line somewhere, we've started to think like scientists. We've started to do experiments, try things out, observe things more carefully, look into mechanisms, trying to understand the mechanisms. We've done this from a starting point that was very close to the naive, innate essentialism that students have, but we never started from exactly that point. We started from slightly off-centre. Now, in the end, we'll have species like fleas and whales that definitely won't interbreed because they've accumulated so many isolating mechanisms. Their genomes are entirely reorganized by chromosome fusions and fissions and other phenomena that interbreeding them is unthinkable. In a way, teaching speciation is like climbing a mountain. You need a good strategy. You could walk along the valley floor, that's easy, until you reach the face at the end of the valley floor, and you're not a gecko, so you can't climb up the wall. You could climb up the scree, but that's going to consume a lot of energy, and we're not moles, so we can't burrow through it. What we would do is we would take a ridge and climb up over the rocks, minimize the patches of scree, and then get to the top. Now, this analogy that I'm using opens the possibility that there are more than one way of teaching speciation. But this shows that there can be a way of teaching speciation and teaching it effectively over a couple of years, practicing the ideas and helping our students to climb that Mount Improbable of the origin of species. Thank you.